So let me introduce Adrianne, who, as she mentioned, is a postdoc in my laboratory. And I'm very excited that she's here. Sorry, my Microsoft Word's just opening. So Adrianne came to the University of Montreal and to the Saint-Justine Research Center from the University of Sydney, where she did her PhD focused on oncolytic viral therapy, so different immunotherapies to treat cancer. She'll talk today, as I mentioned, well, her research now is focused on multiple things, COVID, but also oncolytic viral therapy, different immunotherapy strategies, and also some agent-based modeling in glioblastoma, which I'm sure she will talk about. So thanks very much, Adrianne.
So yeah, thanks Morgan for the introduction. I'm assuming you can all see my screen, but if you can't see the PowerPoint slides, please message me in the chat or let me know. So like Morgan said, I'm going to talk about some applications of computational modeling in cancer. The talk's going to focus around kind of three main areas. First of all, I'm going to talk about agent-based models, cellular automata, off-lattice and on-lattice agent-based models. Also, just a brief discussion around some data optimization and how we fit some models to data, and then looking at different in silico clinical trial techniques, so how instead of in silico clinical trials are created. And at the end, there'll just be a little description about the small MATLAB tutorial we're going to go through after this, which is just going to cover really basic techniques in data optimization and in silico clinical trials. So to start off, I just want to kind of recap again how great mathematics is in cancer research. So cancer occurs at many different physiological scales. You've got like the local tumor microenvironment, if you consider the whole tumor, and the interaction between cells, immune cells, viruses, and so on. And then you can model just the cells themselves if you wanted to consider what happens within a tumor cell or an immune cell or the receptor binding dynamics. Then you've even got the smaller scale when you consider the actual treatment aspects such as chemotherapy and viruses, and you think about antigen and antibodies and cytokines. And some models in mathematics even look at DNA, so how the DNA mutation or mutations in genes might infer resistance, just like Morgan spoke about. And so what I'm going to just go over to start before I talk about any mathematics is a treatment called oncolytic viral therapy. So this is one I do a lot of work modeling in, and I wanted to just recap it in case no one had ever actually heard of it before. It's a really kind of exciting, somewhat promising treatment which has the basic idea that you take a virus and you genetically engineer it so that it will selectively infect tumor cells. So you'll have the virus, it'll selectively infect and replicate within tumor cells. So that means that once it's inside the tumor cell, it replicates, creating infected cells that then burst, releasing thousands of new viruses. And these viruses then go on to infect nearby cells. So you kind of have this round and round process until your immune system gets savvy to what's going on and starts to come in and actually clear the virus. This is a really exciting therapy that lots of people have been talking about. And a lot of the time it's looked at coupled with immunotherapy. And immunotherapy has a range of different treatments that they're looking at, but the idea or the premise behind it is really how can we get our own immune system to recognize and kill off the tumor. So Morgan's already talked a lot about continuous modeling, so I just want to briefly recap that idea before we jump into agent-based modeling. So you've got a few different kind of areas that you can subdivide continuous modeling into. If you think about, say, a well-mixed population where, just as the example we just saw with Morgan, where you've got a population of susceptible tumor cells and resistant tumor cells, and they're interacting in this way where susceptible cells, these tumor cells that should be killed by the drug, become resistant, and that means that their likelihood of death changes. So you can model that simply with equations such as this, like an ODE system. But if you wanted to think about maybe the fact that space might be having a big impact on the tumor response to treatment. So for example, if you think that potentially the spatial arrangement of the tumor cells might be affecting the resistance that's occurring in the population, or the spatial arrangement affects the proliferation of cells, how quickly they're dividing. As we know, within tumors, the center is more quiescent or necrotic, so you would expect that in the center of large tumors, you're going to have slower proliferation. So in that case, another type of continuous modeling that's used a lot is PDEs. where you can model the density of cells spatially within the tumor and how that might affect their proliferation rate or the transfer to this resistant population subpool. So continuous modeling is really great for modeling a lot of different aspects in cancer, but then a lot of people like to turn to agent-based models as well. So I wanted to kind of try and summarize the major overall difference between these two models with this example. So if you think about, you've got a tumor population that's mixed with, say, your susceptible and resistant cells, or just two different phenotypes of cells, and their distribution within the tumor dictates some dynamics such as proliferation or susceptibility to a drug. So this can be modeled as a continuous system where you track the density of these cells over space, such as this X radial dimension. Or if you think about it in agent-based model, what you'd be wanting to do is model individual cell interactions. So instead of modeling the density of a particular type of cell in space, you think about maybe the velocity. So this cell here might move in a certain direction R with a velocity V, or you might actually, instead of having a continuous function for the proliferation of cells, you'd have that there's a probability that a particular cell might proliferate in a certain time step. In addition, instead of considering just a transfer rate between the resistant populations, you might have that cells need to actually physically interact for resistance to occur. So you might need a susceptible cell or a red cell of this phenotype to interact with a blue phenotype cell to then change its resulting phenotype. So this is kind of like a summary or a brief example of the differences that you can see between continuous models and agent-based models. So as an overview, agent-based models in cancer, they primarily consider the agents that they're modeling, so the individuals in the simulation to be cells. But in saying that, there are models that do consider these agents to be humans. So agent-based models, broadly speaking, model individuals, cells or humans, and their interactions with either proliferation, with probabilities or rates, and that evolution of interactions of the population of agents over time. So an example in HPV, HPV is the virus, it's an oncovirus known to cause cervical cancer. So there have been some agent-based models that look at the spread of HPV using an agent-based model to track, say you've got one individual who's infected, they infect another person who then goes on to infect someone else, who then subsequently infects 3 other people. And models have looked at what the impact of HPV vaccination is on this spread in different countries. So for example, if this person did infect someone but was vaccinated, the impact then and the flow-on effect of that person then not transferring on HPV, and then the incidence of cancer after that. So that's one example of when the agents in the model are considered as humans, but most of the time in these models, we think about the agents as cells, and they're usually called cell-based models. So I like to think of it as a subset of agent-based modeling because it's quite a broad field. And within cell-based models, we have 2 types of models that we'll talk about today, and that's lattice-based and off-lattice-based. Oh, this is actually the wrong one. Lattice-based is cellular automata, off-lattice aren't cellular automata, but that'll become clear in the next few slides. So an example of kind of how these models are built can be thought about in terms of proliferation. So in cell-based models, the probability of a cell proliferating in a time step, delta t, can be modeled in a few ways. And I've just put down 3 very basic examples, but there's many others out there. So it could be as simple as considering that there's a fixed probability that a cell will proliferate in any time step. Or it might be that the probability or the likelihood of proliferation is a function of the time since last proliferation. So if T-prolif was the last time that cell proliferated, as that time since the last proliferation increases, the likelihood that cell will proliferate again might also increase. In addition, lots of people look at modeling the probability of proliferating as a function of the distance from the periphery of the tumor edge. So like I mentioned before, with quiescence, we know that cells towards the center of the tumor might not proliferate as often. So a lot of people model proliferation as a function of that distance to the nutrient source or the tumor periphery. So then I want to talk first of all about what lattice-based models are and their comparison to off-lattice-based. So lattice, as it kind of lattice base, as it kind of sounds, means that the agents or the cells in the model are restricted to specific points in the domain or specific points on a lattice. So here's some examples that I think really kind of help to understand what a lattice-based versus an off-lattice-based model is. And I just want to preface this by saying this is a really great review of cell-based models, and I really recommend it if you want more information on this front to go and check out this citation down the bottom here. Oops, sorry. So there's a few types of lattice-based models, but these three kind of cover them here. So there's the first one that I mentioned before, cellular automata, that lots of people may have heard of. Basically, each lattice point is designated a particular cell. So each point in the lattice that's predefined is a cell. And cells move throughout the lattice by probabilities or rules governed by different things. So for example, if a cell proliferates, that might push another cell to move into another lattice point. Or alternatively, if you do have cell proliferation, say this blue cell proliferates, it may put its daughter cell on a nearby lattice point that's empty. Whereas in lattice gas cell automata, what they're actually doing is considering that there's multiple cells at the same point on a lattice. So in a way, it can be computationally simpler or quicker, I should say, because you're going to be modeling multiple cells at a single lattice point and then just the transition between lattice points. But in some ways, you actually do want to consider individual cells at every lattice point. In the cellular pots, it's just an extension of this cellular automata. And instead of every point being inhabited by a cell, you'll have a cell inhabiting multiple points in the lattice. You can see this here, this blue cell is on multiple lattice points. And the advantage of that is it really allows us to capture cell morphology across the lattice. So these are on-lattice agent-based models or cell-based models. Off-lattice is where the cells move freely in space, and movement is governed by a bunch of different rules that I'll talk about in the next slide. Basically, there's a few different types of these models, but they can be divided into center-based and boundary-based. Center-based being where you track the center of cells through the space, and boundary-based where the boundary of the cell is what you're modeling and what you're accounting for as the cell moves around freely in the domain. So here's an example of just the two different types. This is actually a cellular automata for fire spread that I found online, but I thought that it was still kind of illustrating the point about how the cellular automata looks when you compare it, say, to an agent-based model. This is actually an example from Fizzy cell that you'll get to see a lot more of later. But basically, you can see cells are moving freely in the domain. So here's how, in general, or an example maybe, of how cells might move in a lattice-based model. So in each time step, a cell is going to be checked for a few things. The first thing is whether it divides. So it might actually undergo division and then place its daughter cell on a nearby free lattice point. It may actually just move because it might be a motile cell. And again, you'd have some probability that that cell would transition to a different lattice point. The other thing that might happen in a time step for a particular cell is it might die. And so that lattice point actually frees up. In addition, you've got to have, if you want to consider, say, resistance or different heterogeneity within your cellular automata, you're going to have that as it differentiates, it might change phenotype. So that's what you see here. And the last one is sometimes when you run a model, you might want to consider that nothing happens in a time step. So some cells might actually not have to obey one of these rules. So you don't have to proliferate, die, or move. You might just have nothing occur in that time step. And so in that case, the evolution for that cell in that time step stays the same. So you can kind of see an example here where we've got this blue cell down the bottom proliferates, and when it proliferates, it moves into a certain direction and pushes the subsequent cells nearby out of the way. So there are a lot of really great models in the literature that have done an amazing job at using these cellular automata in much more complicated ways than what I just presented on the previous slide. And so I'm going to do my best to give them justice, but I do actually recommend, if you're interested, going and looking at the references down the bottom to get a better understanding of what they did. So here what the authors did is they looked at this tumor growth paradox. And so they created a cellular automata where it obeys rules just like this here. So you've got, say, you consider cells, they increase in age and they see whether they have space. And like I said, if there's not as much space or they're in the center of the tumor, they might go into a quiescent cell where they don't proliferate. Then you've got the option that cells might migrate and mature, they can undergo death. And if they undergo death, is there any space? And so on. So this type of tree diagram or schematic is a real good representation of how you build a cellular automata, where you have rules and ways of transitioning between the different stages in the model. And so they again have a movement similar to the one I showed you on the other slide, where if you've got empty space around you, you can move to any of the empty spots in the lattice. However, if you're crowded in like this, where there is no space around you, you won't actually move. So the really great thing about their model was that they were able to show that as you increase the cell death rate, that what you see is that initially in red is no cell death, and as you go down, it's faster cell death. So initially you're disadvantaged as a tumor if you increase the cell death rate, but overall you start to really outgrow the rest if you have a larger cell death rate. So you can see that here, when there's no cell death rate, a small cell death rate, and quite a large cell death rate, you can see the tumors really, really evolved. Okay, here's another just quick example of a lattice-based model. Here they designed a Voronoi tessellation as their initial tessellation, which I'll talk about as their initial lattice, and I'll talk about that in a few slides. Basically what they wanted to recreate was the GBM invasive tumors that you see down the bottom here. So you can see this is a snapshot of kind of a short time evolution where you've got your red cells. The red cell proliferates into a green cell, which becomes an invasive cell, which means it can break down ECM and move through the lattice. And you see that happening here where the invasive cell is moving into the lattice, leaving behind blue cells. And so they were able to look at homogeneous ECM and its effect on the evolution of an invasive GBM branches, which you see here, and random ECM and how that affects the invasive GBM's branches that it forms. So again, really great reference to look at if you're interested in learning more about cellular automatas that are in the literature for tumor growth. So off-lattice models are different to the on-lattice models that I spoke about before, primarily because, like I said, they're not confined to a particular lattice or spatial arrangements. They move through the space. And how they usually do that is by solving force equations. So the one that I'm most familiar with and have used in the past is Hooke's law. So you might remember Hooke's law from, if you did an undergrad, like a Hamiltonian Lagrangian course or mechanics, but basically it talks about how spring contracts when you have a kind of a rest length for a spring. If you were to pull it and push it in, the behavior it would have. So that's described here by Hooke's law. So if you imagine you had a cell K here and it proliferated into two daughter cells, and all these cells were modeled as connections with springs, you can imagine how the system would rearrange from that. Another option, if you're not going down the Hooke's law path to model this, is to actually consider the forces that are acting on cells. So for example, in Fizzy cell, this is a simplified version of the model in Fizzy cell that Paul and Randy will talk about more. But basically, if you had two cells and they had attractive forces and repelling forces, so forces that were either keeping them together or pulling them apart, and then the cell was also being affected by locomotive, so it had a direction it wanted to move in, whether that was just random or by chemotaxis. And then it also had a drag force on it. So it's maybe in a fluid and it's feeling some sort of drag through the environment. And so you can model that type of interaction by this. And that's how you update in every time step your cell movement and where it would go, by calculating the force felt by that cell. So if I go here, here's an example down the bottom here, which is quite small, I know, of this Voronoi tessellation with Hooke's law. So Hooke's law being that one I spoke about in the last slide with the spring. You can see in green the tumor forming in the tissue here and pushing the nearby healthy cells, which are in brown, out of the way. So this is, again, an off-lattice model, even though it looks like it has a lattice kind of fixed configuration. The Voronoi tessellation is just imposed on top of cell positions. So a Voronoi tessellation, in case you haven't heard of it, is a really interesting tessellation. It basically is when you put points in your space and draw lines that are equidistant between any 2 points. So for example, this line here is all the points in this space that are equidistant between these two circles. And so with these types of models, we've investigated oncolytic virotherapy that I spoke about earlier and the effectiveness it has at actually treating the tumor. So you can see in blue the virus infiltrating and killing off this aspect of the tumor, but you can also see the tumor is actually escaping away. And so we've used these types of models to investigate where should these initial injections be placed. And so I'll do a brief example of some work that we've been doing in Fizzy cell with glioblastoma. So here what we did was create, we recreated actual patient slices. So this here and here is from a single patient tumor. This is one aspect of their tumor and another. Pink is the stromal cells and purple is the GBM. So you can see in this sample here, it's got quite a lot of stroma, whereas in this one, there's a lot more GBM cells. So in Fizzy cell, we recreated that same density in a random way, but at the same density ratio of stromal cells to GBM. And then we created a model for how that tumor might actually perform either this sparse aspect of it or this dense aspect under virotherapy. And so there we go. So basically what happens, like I mentioned before, is viruses infect the GBM cells and turn them into brown cells. And this cell then causes an immune response, which I haven't really spoken about a lot before, but if you're interested, basically we consider that there's T helper cells and CD8 cells that are within the tissue and contributing to infected cell clearance. And so you can see in animation here, the virus is infiltrating. You don't actually see the virus per se, but you can see the infected cells in the sample. What was most interesting about this model was that we were able to replicate what they were seeing in their actual tumor slices. So they took those same patient tumor slices and actually tested the virus on them and saw this kind of irregular infiltration pattern. And what we'd started to be able to do was actually replicate that with this Fizzy cell model. So now I've kind of talked a little bit about off-lattice and on-lattice agent-based models with some examples. The next thing I wanted to discuss was data fitting. So the goal a lot of the time when you're trying to fit, well, when you develop a model is to try and get realistic parameter values. And so when you're data fitting, what you're trying to do is find model parameters, P, that minimize the residual between the data and the model. So you can see that here with this purple line, let's say this was your model evolution. Just here, YT I of P would be each point on the model that corresponds to where there's a data point. And the residual is just the difference between the data and the model. So we really just want to find a parameter set P that minimizes this difference. And so least squares is one that we'll look at later in MATLAB, but basically looks to minimize the L2 norm. So it's taking this residual and then just squaring it and summing them all together. And it looks to minimize that particular value. So what P values will minimize that? And there's a few different ways that people use to look at this. One is simulated annealing, which is pictured up here, where your initial guess, you kind of you might find a local minimum, but simulated annealing really tries to find a global minimum through perturbations in that parameter set that it's trying to minimize the distance. And another example is gradient descent that looks at finding the gradient that descends through your surface or your model's kind of surface for all the parameter values. So I wanted to also just briefly discuss what you should do or what people can do or have done when they have multiple data sets. So here's an example of a simultaneous data fitting. So we had a treatment where you had just a virus that's called an adenovirus that they measured tumor growth under. They also measured it under a pegylated adenovirus and a pegylated adenovirus with Herceptin. So you can see they've conducted 4 data sets, 3 with the different types of virus and 1 that was just tumor growth alone. Now with this, since there are 4 data sets and they have this same virus just with different modifications, instead of fitting the same model just separately to each 4 data sets, one thing you can do is do a simultaneous fit. So it's where you fit all data sets and all individual data points to the model that you might have for the infection of the virus and production of new virions. And so that way you can obtain multiple parameters that talk to 4 different data sets and then just have unique parameters for the difference between the data sets. So for example, in this case, we found that the infection rate, so how each of these viruses was infecting tumor cells, could be the only difference between the data sets. So in this way, when we fit all of these parameters, but most of them were fit at the same time to all 4 data sets. But a different example, instead of doing all the data sets at the same time, is to break down the experiments in the data sets and do sub-model fitting. So here's kind of another example, like an applied example, where we've got a virus that's expressing these 2 cytokines, IL-12 and GM-CSF, and it's also put into a treatment with dendritic cells and loaded onto a gel. So the experimentalists when a lot of the time they do these experiments that are in stages. So you've got a control experiment with no treatment, you've got a treatment just with the virus, a treatment just with dendritic cells, a treatment with the virus and dendritic cells, and then a treatment with virus, dendritic cells, and gel. And these are like the jackpot, I think, these types of experiments when you're trying to fit and model, because it means that you can fit, you can fit stages of your model to each sub data set, and that's what we do here. So for different parameters in the model, you might only fit one data set, and then you fix it for the remaining data sets. And so that's an example of hierarchical fitting. And so the last thing that I just want to give some examples of again is in silico trials. So the idea behind those is to use a lot of the methods I've already spoken about, either agent-based models or continuous models, fit them to data. So you develop a model and you fit it to data. From the data, then you infer some identity about a cohort of patients, and you run a bunch of treatments on them to see what might perform best. And then you look at what happens once you implement that treatment. So here's an example. Morgan already spoke about it, but I also just wanted to kind of reiterate it in like a flow diagram so you could see the general process of building and running an in silico trial. So first thing, of course, is develop a model. So this is from this paper down here, which again, a great read if you want to know more about how these are built and run. So they built a model to look at an oncolytic virus TVEC that's been approved by the FDA. The FDA for melanoma, and the combination of that with GM-CSF, the type of cytokine. They developed a model, fit it to data. From the data, they then generated distributions of possible parameter values and sampled from those to create their patients. So this is actually the resulting distribution of a parameter value for the cohort. And you can think of it like a characteristic distribution for this biological parameter that has a meaning. So with this, they then had 200 patients that they could actually simulate treatment on, and from that determine some optimal therapy. And so what they were able to show, which I know Morgan's already mentioned, but I'll just reiterate, is that they showed that the standard combination therapy has a survival curve down here, so you can see it just here. But with their clinical trial cohort and their protocols they tested, they were able to find a protocol that was better, or at least that their in silico trial suggested was better than the standard combination therapy. And the advantage of this is that you don't actually have to run anything on any actual humans or animals, because you can test all the possible options first in silico and then join that up to a clinical trial. So for the sake of time, I won't go through that example. I'll just go through one last example. So in the previous one, I said that they took a model and fit it to data and used the data fit to then infer the distributions for their parameters for their patients. Another option that has been developed by a different group of people was, instead of to fit to the data, to use the data to infer a bound on responses of the patient. So here is a tumor volume. You can see it here, the mean and standard error over time. And then what you do is you take the bounds of the standard deviation, and you make that your kind of area through which patient growth must be within. And so with this area, you then, first of all, sample from a uniform distribution of possible parameter values. You simulate the model, which in this case, let's just say is simple tumor growth. And then with that simulated model, you then look at, does the patient actually sit within these bounds? And if not, you run an optimization routine like simulated annealing until your patient is within this region here, and then you accept them into the trial and continue on. So one thing that's another really important aspect of cancer modeling, computational cancer modeling, is how you optimize treatment protocols. So once you have a cohort of patients, how do you then determine or evaluate what the optimal treatment protocol might be? And so one example is a genetic algorithm, but there are many others. So some people choose to just numerically simulate all possible outcomes, which can be computationally expensive depending on how many treatment options there are. There's genetic algorithms, gradient descent, and simulated annealing. So genetic algorithms, I'll just go through as an example, but a lot of them are similar-ish, but have different methods for how they actually get to that optimal point. So a genetic algorithm, what that looks at doing is creating a random initial sampling in your parameter space, and each of these parameter sets would be a parent. So what happens is you get this kind of nice random sampling around an area, an interval in your parameter space. These parents then all have offspring or create children. So they pass on their parameter values to their children in a few different ways, either through a crossover, mutation, or direct descent. That's all based on the fitness of your parameter space. And so eventually over time, you can see as these new kind of generations of parents are chosen, you start to converge on the minimum in your space. So I think that I might summarize and leave it there. So I know there's a lot of information there. Some of it you might know, some might be new, but I hope it's given a kind of general overview of the techniques in this area. So in agent-based modeling and data fitting, and also in silico clinical trials, a lot of this agent-based modeling you're going to see in about 40 minutes with Paul and Randy. We're going to take a break, but when we come back in, say, 10 minutes at 1:43, we're going to do a short MATLAB tutorial that's going to look at how we might fit to data and then how we might run an in silico clinical trial.
Before we break, sorry, I'm going to be that person. Thank you very much, Adrianne. That was great. There's just one question I think maybe you could deal with quickly, and then we'll take a break, which is, are the time steps in lattice-based models the same in simulation world, equivalent to the time dynamics in the real world? So a lot of the time what you're doing when you're creating a lattice-based model is you're defining a time step which you then can correlate to a real-world time step. So I can't see the question anymore, but yeah, basically the models are built so that we can then correlate them to the real-world time step, so they actually have a real-world application. So yeah, yes, I think the short answer is yeah. Great.
I will look at the questions. I think I see there's some questions. I will make sure to go through them now in the break.
Great. Okay. So let's come back.
Why don't we say 1:45?
Yeah.
Great. A lot of details for what I'm about to code up, just so it hopefully makes a bit of sense. So there's two data sets, like I mentioned, a control, which is only going to have tumor growth in it, and an immune one, which is going to have tumor growth and T cells. So we're going to call this data set 1 and data set 2. And now they're going to have different models that they speak to. So since data set 1 has no T cells, we set T cells to be 0 and just consider tumor growth. So just logistic growth here. And we're going to look at minimizing these two data sets or fitting them simultaneously, so at the same time. So if I jump into the MATLAB, the thing you're going to want to open up, I go into my folder, is the code called commands. So I've basically filled out the code, but hopefully if you hit run, it shouldn't work. Oops, mine worked. I've gone into the wrong one, sorry. I've opened the one that does work. If you open your code though, hopefully it shouldn't work if you hit run. Yeah, it should give you an error like this. So basically it shouldn't run because I've removed a bunch of stuff from it that we need to fill out. So as I'll go through and explain it. So basically we're loading in the data up here. So I've already pre-saved it as a map file that I'm loading in. And the data is saved like this. It's saved as experiment time, the control tumor volume, the control tumor volume standard deviation, the tumor volume mean under that immune treatment, and the tumor volume standard deviation. This is meant to be standard deviation, so you're going to want to correct that. The tumor volume standard deviation under treatment. So you can see there's the two, if I flick back to this one here, there's that control experiment, the time for both of them is one variable, and then you've also got the tumor growth mean and standard deviation for the control, tumor growth mean and standard deviation for the immune treatment. So make sure you change this to _ standard deviation if you're following along. The first thing that we want to set is an initial tumor size, so how many initial cells there are. And so you can see on the data graph down here, it's roughly 100. So we're going to set that to be 100. We're going to say that there are no T cells initially in the immune experiment because we're assuming that they're becoming activated by the presence of the tumor cells. We then also need to think about what the growth rate parameter should be, so what R should be and what K should be. So for now, let's just assume that our guess of 1 for R might be enough. And we need another guess for K. So K is the carrying capacity of the tumor. So basically, how large is it going to get before it stops proliferating? And so it's definitely going to get above, you would say, 5,000 because it's not there yet. So it might get up to, say, let's just say 10,000. So we'll set R to be 1 and K to be 10,000. And then I've already set some initial guesses for the other parameters. So if I just switch sides here, you can see this killing rate kappa, I've set an initial guess of 0.001. And for the activation rate and the death rate, I've just set 1 and 1. So what am I doing here? Well, I'm taking the parameters in the model and I'm giving them an initial guess to send the fitting function. So remember, we're going to fit this actual model to the data, but you need to start off with a guess for the fitter function I'm going to use. So now that we've initialized the variables for the experiment time, mean, standard deviation, and so on, and we've also initialized parameter values and the parameters that we're going to fit here, we then have to actually run a fitter. So if you now open up the code simultaneous fit, this one here, it should look a bit like this. Sorry. So what we're basically doing here is this code is actually a fitting function that I've written, but really I have just taken the parameter values that I set here and I've put them into this structure called P, and the data that I've popped up here into this structure, and I'm feeding them through to the function simultaneous fit. And so they're coming in here. Now this code here, what it does is it takes in the parameter values and the data that we've preset, and it tries to fit them to those data values, that data that I showed you on here. You can see it here. So the first thing we need to do is initialize our parameter guess vector. So like I said, we're going to make guesses for these parameters here, but we actually need to initialize that parameter guess vector. So let's just do that. So parameter guess would be p.r, so the r parameter, the k parameter, the kappa parameter, the a parameter, and the d parameter. Now I'm fitting all of these parameters here, but realistically you probably would only want to fit maybe 1 or 2, depending on how much data you have, but I'm just going to fit them all today just to show you how it works. This line here is just simply, this line here calls the fitting function that's inbuilt in MATLAB. So it's called LSQ non-lin. There's a few other fitting functions I mentioned in the slides, but this is the one we're going to use today. So basically, this parameter guess is the initial guess for LSQ non-lin, or the fitting function, the least squares nonlinear fitting function. And what it's going to do is it's going to take the parameter guess and feed it into this function called residuals function, which is down here. So remember, our goal is to find a set of parameters, these ones up here, that minimize the difference between the data and the model. So to do that, what we need to do is, first of all, take in whatever parameters the fitter is sending and initialize them as the ones we're trying to fit. So R, K, kappa, A, and D. The next thing we need to do is actually then evaluate the model for these parameters. So to do that, we have, like I mentioned, 2 models we're considering and 2 data sets. Just a simple control with just tumor growth, and the tumor growth under an immune treatment, so with a T population. So the first data set is just going to be the residual. The first thing to do is just to calculate the residual between the first model, so the simplified tumor growth, and the data. So remember, this is the control data, and this here calls the simplified model down here, just here, which is just logistic growth. So no T cells. I'll move that over. So you can see here, no T cells, just logistic growth. And it solves it and matches it to the data and calculates that residual, so the difference between the data and the model. So what we're going to add in here is the second one. So that's the first one. We need to actually write the second model down. So you want to write your initial conditions for the second model. Well, that's just this full model here will be S0 and T0. We're then also going to want to solve the second model. So let's write that down here. So we just copy this line. And in this case, we're not going to solve the control model because we've already solved that above for the first data set. We're going to solve what's called the full model, which we haven't written yet, but we'll write in a second, which is just this full model here. We're going to solve it from 0 to the end of the data times, that's fine, and we're sending it these initial conditions. And then the last thing we're wanting to do is calculate the residual, which is the difference between the data and the model. So it's going to be the model evaluated at the data. So what this does is it evaluates the solution to your model here at the data.time, so at the time points of the data, and we're going to minus it from the treatment data instead of the control. So remember, we're doing this data set here. Now, what we actually need to do though is actually write this model. So to write it up here, what we do is we first initialize the two variables s and t. So s is just y of 1 and t is y of 2. That's because what this is an ODE solver, and that uses a variable called y. So we need to actually put that in terms of our notation. And then we just copy in these equations here, which is our full model. So r times s times 1 minus s on k minus kappa times s times t a times, oops, sorry, so just dt equals a times s minus d times t. And then the last thing we want to do is send the solution or these ODE forms up here. Just so you know, everyone, I will be sending the full code around at the end of this. So if it's not clear what I'm typing, because it's a bit zoomed out, I'll make sure to actually share that with you at the end. So you can follow along if it's possible. Otherwise, if not, I'll absolutely send that to you at the end. So now we're solving both the first model fit to the first data set and the second model, which has the T cells with the second data set. So that's how, and that's the fitting function. So remember, this was actually the fitting code we're calling. So it's going to calculate this residual for its parameter set guess, and then it's going to keep iterating through this until it reaches a minimum. So ideally now, I don't know if I can make the text a bit larger. It's a great question. I'm not quite sure how to do that. It's probably a way. I'm not sure, but I will definitely share the code at the end. I'm sorry, it's so small. I will, yeah, sorry, I'm reading your comments now. I'll definitely, yeah, I wish I could make it bigger. There's probably a way, but for now, I'll just, this home preferences font, thank you. Oh, in the main toolbar. 1 second. Oh yeah, it's set at 10, probably too small. That must be that would be a lot better. Yeah, sorry about that. Thank you so much for that. Okay, I hope that's a little bit more visible now. I apologize, it was extremely small. So basically now we've made some changes in this main code, and if we hit run section, ideally we don't hit any errors. So if you open up the command window after, so run section, if I go back here, run section just means that whatever's highlighted in yellow is what gets run. So if you run this section here, when it actually has all the code filled in, you end up with this, this response here, which just says that a local minimum was possible. And so LSQ non-len has stopped. And this is the fit that we get. So you can see here that in blue is the data and in black is our fit. Now remember, we actually fit 2 data sets and this is only one of them. So we want to actually do the second data set plot as well. And so if you scroll down the bottom here, you can see this was our fitting function. You might remember I mentioned earlier. So basically we set our parameters, sent them through to this fitting function, which I then opened up and changed and added a few things to. And then we come out here and it's plotted one of the data fits. So if we want to plot the second data fit, we just need to repeat this up here, but for our second lot of data. So what we had here was the time and the control data, but we're also interested in the treatment data. So remember, there were two data sets, a control and a treatment, and we fit both of those to the submodels. So now we want to plot this. So we want to plot data.treatment and data.standard treatment. So replacing the control down here with data.treatment and data.standard treatment. And also we need to now, that will plot our error bar of the data for the treatment data, not the control. And if we also now want to plot that against what the model comes up with, we want to plot that with the full fit, not the control fit, but the full fit. So we want to change this word to full and this one to full as well. And so if we now, yes, I can pop that in right now. So great suggestion. Lizette's just said, why don't I just share the code with you? And I think that's a great idea. Let me just do that right now. So I'm just going to upload it onto the Dropbox link. The Google Drive link that I sent above below. Hold on one second. So I apologize, everyone, that I definitely have gone too fast, and I think that my text was too small, which was not helpful at all, and I am very sorry for that. So if you go into the Google Drive, which I'll post the link again down the bottom, just there. So that's the link in the Google Drive. If you download the code for tutorial that says working, it has everything filled in and should run when you hit run. So please let me know if that works or not. I apologize again that it hasn't worked as well as I'd planned. Still new to virtual workshops. So just continuing on, if I run this full section, this is our fitting section again. I just want to show you the full results of the fitter. So what happens is you get these two fits here when you run it. So this is our control experiment. This one says control, but it's just because we haven't changed the title label yet. So if you change that to immune treatment, and we just run this line by pasting it in the command window, you get the control and the immune treatment fit. So you can see that this code definitely, once you've downloaded it and actually got to run it properly, will actually give you fits to those two data sets, here and here. So that's kind of the very basic kind of rapid overview of how you might undergo fitting with these. I'm always happy to answer any questions anyone might have at any point that are about these. Obviously, this has been very quick and not one that you could have typed along with necessarily. So I'm very happy to, at any point, take anyone through this. With the last few minutes remaining, I do want to show you, just so we can see how the code works, so that on your own you're able to run it. Just so you know, if you were to download the code and if you hit this full run button, it will take a while. That's because it's actually running a genetic algorithm as well. At the last stage, it runs a genetic algorithm, and that actually takes a while. So if you do want to run it, I suggest running it by sections. So you can see the fitter first, and then you can see a virtual patient cohort being created. Okay, so someone has just said does need a global optimization toolbox. I was worried about that, and I do apologize. I think that's something you can opt to download in the installation of MATLAB. I think it's just a box you might tick, but you might need to look into that, whether it's a version thing or it's just when you download it, you might have to actually select global optimization toolbox as well. So for the last 5 minutes, I just want to show you basically an overview of how we can generate a virtual cohort and then what it looks like. So the first thing is we've got our fitted parameters. Hopefully, if you've downloaded the real code, you do have them as well. And so we've got a set of fitted parameters. What we're going to do now is create a normal distribution around those fitted parameters with variance 0.1 times the mean or the fitted parameter value. We're going to sample from it. And then we're going to make sure patients tumor volumes sit within the ranges of the data. This is just a repeat. It should be the other data set. We're going to make sure that they sit within the ranges of the data by solving this equation here for G. So this basically says that the difference between the patient's tumor volume and this data point has to be within this shaded region. So doing that, if the patient is successful, you can kind of see one here. This patient was successful because I've said that we're allowed to be 3 standard deviations from the mean, and this is just 1. So you can say this patient here, they had a unique parameter set and they were successful. So they get added to the cohort. We keep simulating until we get the number of patients that we're after. So if I'll open up the working code because that's probably what everyone's following along with now. So if you scroll down to the section that says creating virtual patients, I've said that the number of patients we're going to create is n equals 50. So that's 50 patients. Of course, 50 is quite a small sample size. You probably want more like 200 or more. And then what I'm going to do is create these normal distributions. So if I show you this again, so basically I'm going to create these normal distributions. They're going to have that their mean is the fitted parameter value. So I'm just going to say R original or K original to mean the mean of these normal distributions. And they're going to be those parameter values we obtained in the fit. And then what we're going to do is we're going to sample. So we're going to try and create N patients. So while the number of patients created is less than how many you want, we're going to keep iterating through this loop here. So we're going to sample from the normal distribution that has, and this command here just is a normal random variable creator in MATLAB. So we're going to sample from the normal distribution that has means as the fitted parameter and variances as 0.1 times the parameter value. So each of these new parameter values is randomly chosen from these normal distributions. And then, like I said, we're going to make sure that their tumor volumes sit within the right ranges that match our data set. And then if they do, we will keep them. So if I hit run, it didn't work. Give me 1 second. It should have worked. Hold on 1 second. So it didn't work, which is just unfortunate. So question, that's a good one, is, oh, it's all right, Morgan's already responded. So it didn't work, but it definitely did before. So there's probably been a small bug that's come up in the model, but I will investigate and get back to you and post possibly the working code in there because it should be working. It worked just an hour ago. Give me 1 second. Not sure, but apologies on that front because it should have worked. Oh, is it working for you? Oh, that's good. Okay. Did you, well, I'm not quite sure what's happened on my end. I'm sorry. That happens sometimes. Yeah, good idea. So I'm just going to clear the environment and just try it one more time in case it was just a sampling area. Yeah, many colored curves. All right, so Gary got it. Gary Wilkins got it. I apologize. Oh, there we go. Thank you. Cool. All right, yeah, it did work. So good, so the working code does work. It just didn't work on my code. So when you hit run, what you should see is that the simulator is plotting many, many colored curves, and it looks a lot like this. So each of these curves corresponds to a patient's tumor growth that fit within 3 standard deviations of the data that we were looking at. So if I go back to this slide here, so each of these lines here, each of these curves actually sat within 3 standard deviations of this data. And so when it did, this patient was accepted into the trial And we kept going until we reached 50 patients, because that's how many we were sampling. And this is the dynamic output for this. So this would be the control tumor growth for all the patients. And this here is the immune tumor growth for all the patients. So obviously there's many different ways to generate these patients. This is just one of them. And I hope an example code that helps you kind of start to think about how these can be run. They're not too difficult and too much of an extension on your generic kind of creation of a model and a fit. So I hope that helps. The last part of the code is simulating a genetic algorithm. So basically what it does is it takes these patients and it simulates if we were to impose a drug on them here, and what would be the optimal drug dosage for the cohort. So if you want to run that, you can. It takes maybe about half an hour to give you an output, because like I mentioned before, genetic algorithms can be, in some cases, computationally expensive when you have a large cohort of patients you're trying to optimize for. But I can show you what it looks like in the end when you eventually get it out. It looks like this. So this is basically all of your patients, what their tumor volume was at day 20 after treatment with a drug. So again, sorry, I guess it's been a little bit of a roller coaster ride with this. I apologize. I'm going to again share that updated link for the working code, and I hope that helps. And please, at any point, reach out if you have any questions. If you'd like me to go through this with you at some point, I'm more than happy to. And thank you everyone for listening, bearing with me, and taking part.